Sir, did we have the class yesterday?
No, no, we don't have. We have just declared in the group, sir. We don't have the classes
Okay.
today. So today,
Fortunately, I mean, I could.
today it will continue, sir. Today it will
Okay.
continue.
Okay. Thank you.
I think this much people only will come as we are recording the session. I think Dr. Vijana, we can start.
Okay, sir.
Yes, yes.
So, good evening, everybody. So, uh, have you downloaded that data what I said in group?
Yes, ma'am.
So everyone downloaded, right? Hello. Okay. So please reply everybody because I'm starting with that workshop. First up. So what I yesterday I discussed about some stuff, right? And these analysis steps. Pipelines. So uh, that first step is like quality control. So I'm starting with that. So without downloading data, it's not possible to you people to understand. Okay. So okay, uh, sharing my entire screen here. Okay. So are you able to see this page, Galaxy page? Hello.
Yes, ma'am.
Okay, ha ha ha. So this is the data like sequence I downloaded from. Parent data I downloaded from Galaxy itself. So uh, here you can see like two parent sequences are there. So here you can check. You can view the sequence. So this is the FASTQ format. The sequence is like the FASTQ format. So FASTQ format means it stores. The sequence that stores sequence information in a plain text format where each read is represented by four lines. Okay. So these are the four lines: one, two, three, and four. Okay. So the first line begins with a, this first line. This first line begins with a symbol at, okay, at symbol, and it contains unique identifier for the read. Okay. The second line, that is this line, contains the nucleotide sequence of the read. Okay. The third line, it's contain the plus symbol, and optionally may contain additional information about the read. And this fourth line, this line contains the quality scores of the read, which reflect the confidence of the sequencer in each base call. The quality scores are represented as ASCII character. So you can see here, these are the ASCII character. For each score, it have a number. The higher Fred score will be the, like we can say, if it is if we are getting a higher Fred score, the sequence, we can say the more confident in the sequencer is in the base score. Okay, then we are okay. First we downloaded, we uploaded the data here. Okay, you can see here 1 2 3 4, these are the data. Then we are checking the quality of the read. So using quality control tool, that is FastQC. So here we are using FastQC. So here I already done FastQC. So you can also start to do with me, like then I think it will take some time to finish the maybe some people they may be knowing that this, and some are maybe new to this, so you just do with me. Then anyway you will get it will take time. So fast QC means it's a it's a tool, like it provides a simple way to do some quality checks on raw sequence data coming from high throughput sequencing pipelines. Its reports mainly uses a traffic light system to indicate whether your results are good or bad. Okay, so you can search here fast QC. Okay, please do with me. So QC, then it will show here, okay, in left side. So click that, then you will get here, like you will get here this type of page. Then you can add the like reads here. So 1 2 3 4. Okay, then these and all you can add like default parameters. If you know, like if you know the adapter list, you can add here, you can select here. But if it is not the, just put it in default default parameters, then you can go for run tool. So the green color, okay, you will get this green color. So it stated like it started the tool successfully. Okay, the green color represent the started tool first QC successfully. So here you will get this side. So this color represents it's in queue. So it will take some time to finish. So after finishing, you will get a green color. Okay. So everybody is following me, or hello?
Hello, I have a doubt. How to
Okay.
select these four sequences together for this?
Control, control, then enter that. Plus control,
Control,
then you just select that sequences. Okay. I'll go one again. Okay. Fast QC. Okay, I'll select here. Then you can go here. You can click here, multiple data set. Okay, select
Okay.
here. Then you will get a sequence here. Okay, our sequence here. So control, then click here, plus control, then click here, then you'll get, you can select this. Could you able to do that?
Yes, ma'am.
Okay, then you just go for default parameter, then run tool. Okay, so everybody following or not? Nobody is replying me. Hello.
Ma'am, kindly can you repeat, ma'am? Because due to network, I left it, ma'am. Again, I joined.
Okay, first you go to Fast QC in tools. In left side, you can see tools, right? So in that, you just type Fast QC. So first we are, after downloading the data or after uploading the data, first we are starting with quality control. So yesterday we already discussed about steps, right? So first step is like quality control. So I'll show that. If you have any doubt, I'll show that. Yeah. So here you can see these are the RNA-seq analysis workflow. Okay. So here is the general workflow. So first step is quality control, then alignment, then quantification, then differential expression analysis, then functional analysis, and after that at last visualization. Okay, visualization of the result. So here in the first step we already uploaded the data that already over yesterday itself, last day itself. Then today we are starting with quality control. Okay, this first step. So for quality control, we are using FastQC tool. Okay, so type FastQC here. You will get FastQC here. Then click it. Then it will open like this. You can see the page like this. Then you just upload. You just click here, multiple data set, and control select. Control and these four set. Whatever is your data, you can select here, and then other parameters you can set it as default. If you have a specific research questions or specific research uh, program, then you can go for this. You can add some parameters here. So otherwise, you can put it as like default. Then you can run the tool here. Okay. If you are running the tool, you will get a green color here. It means the tool has started successfully. Okay, so here in this history lane, you will get this gray color. So it means it's in queue. So anyway, it will take some time to finish. So today you will get maybe, but it will take time. So for that, I already did the analysis and kept aside. So I'll show the result how it will be. So this is the my past QC result. Okay. fast QC result. This is my fast QC result. So it will contain like raw data. The result will contain for each sequence. It will show raw data plus web page. Raw data means you can see here. Raw data. It will be like this. You will get like this raw data. It's a text file. Then a graph and report. The side you can see summary. So here everything like okay, this I'll come like this. So here you can get basic statistics of this report. So here you can see file name. So this is the file name of that this one. Then file type, that is conventional base calls. Then encoding, so it is the Sanger Illumina sequence, so it's showing here. Then total sequences of that tree, so total sequences. Then sequence flagged as poor quality 0, so our quality is okay. So sequence length is 75 and percentage of GC content is 48. So almost 50 percentage we have a GC content. So here by clicking here you can see each graph. Okay. So green color represent it's okay, like it's very good. And yellow color like, so we have to check this. And red means it's in any way it's very wrong. So by clicking here you can see each graph. Okay, everything you can see here. Duplication levels, our representative sequence, adapter content, everything you can see here. So uh, okay, in this graph you can like you can see this blue line. Okay, so the blue line represents average quality scores here. Boxes. Okay, yellow color represent here you can see green color represent so this green color represent, this green color represent very good quality. And the quality here you can see the quality uh, is okay here. And again towards the end again quality is decreasing. Okay, so if quality is not good, we can trim the sequences and discard the poor quality regions. In the next step, we will be seeing that how to trim the data. I mean, how to trim the trim the sequence. Okay. Then like here okay here okay.
Is it my problem or Vijayana's network is lagging? Or my problem? I don't know.
I don't know, sir. I don't know, sir.
Everybody
Okay. This
very lagging in the network? No. Maybe my problem. Okay.
okay, maybe.
Okay.
Okay, sir. This is the distribution of average quality reads, and here the x-axis represent the average quality scores as it moving to right. Quality score increases, and the y-axis represent number of reads. Okay, these of each average read. So we want all our reads to be uniformly distributed, and here you can see this is not that much bad. Okay, this is okay. Quality content per base sequence content. We are checking this. So here you can see yes, sequence content. Okay, okay. So here the distribution of different bases at each position along with the read. Okay, so here you can see different colors. Okay, so here x-axis different colors it represent different bases. Okay, so x-axis represent the position, x-axis represent the position, and y-axis represent, here y-axis represent the percentage of different nucleotide bases identified with expect the line almost parallel. This one is not good. So normally what we expect is like it should be like almost parallel. So here so many you can see this graph, right? It's not parallel to each other. So here something is wrong. So after trimming it will be good. So then we can check per sequence um, GC content. Okay, here you can see this graph. Okay, this is mainly the plot. Blue line represent normal distribution. This blue line represent normal. It should be like this. I mean normal distribution, and the red line represent actual distribution. Here also something is like wrong. So we should be sequence duplication levels. Yeah, sequence duplication levels. So here you can see sequence duplication, how much it will be. Okay, so this is the graph. Okay, so here blue line says total sequences, and the other one is like you can see here it will be the blue line is like total sequences, and red line is like duplicated sequences. So here you can see uh, the how much duplication happened here. Like look at the blue line and red line. So you can see how much duplication. Here you can see this portion above like greater than 10, it's duplicated. Almost duplicated. And here you can see more duplicated.
Telling that blue also that the same sequence, red comes
That is different diagram, right? Here it's like that. Yeah. Here by looking up to 10%, it is duplicated like 2 to 3 times. 2% like that. So here you can check like this here you can check like from here to here you can check it's containing duplicated more than or more than 50 percentage. Okay,
Good.
not 1k one time, it's time,
More
more
than
than 10
500.
times. Sequences in the sense base pairs, base that bases is
Number
duplicated.
of sequences.
Yeah, yeah.
Ma'am, then how much level we can accept, ma'am, in this level duplications and all? Can you tell me?
Normally it should be like that bad, but somehow in this case it is good. I mean it is okay. Like adapter content is okay. It's showing green color. This much level is okay. I mean if it is like more than 50% or more than very this one, so it will be affecting that I mean result expression levels. So we should be I mean after trimming but here it is okay, but after trimming also you can check the data, then if it is again it's showing wrong, then you can cut again.
Ma'am, how to trim?
Trim I'll show now. Just I mean second step is like trimming the data. So for that, okay, next step we can trim the data. Okay, so for that we are using Trimomatic.
Excuse me, ma'am.
Huh.
What is the acceptable level of duplication increase of transcriptome data?
That I'm not sure. I'll share in group.
Oh,
So
okay.
normally, okay, I forgot actually that number. So maybe I can share in group today
Okay.
itself. Okay. So next we are going for trimming. Okay. So normally first you can trim like before first QC you can trim the data and then check first QC. Otherwise you can check the first QC first. I mean quality first. Then you can go for trimming. Okay, so both are good actually, both are correct. So but that is depend upon your this one pipelines. Then first you can go Trimomatic, then you can check the quality. Otherwise you can check the quality first and you can trim the data. So here we are doing first we are checking the quality, then we are trimming the data. Okay. So here we are using for trimming we are using Trimomatic. Okay. So many tools are available. That's everything is depending upon your research needs. Okay, research questions. So here in this case we are using Trimomatic. Okay, Trimomatic means it's a popular bioinformatics tool for trimming and filtering next generation sequencing, that is NGS data. Then it is mainly used to remove low quality reads, adapter sequences, and other contaminants from NGS data to improve the accuracy and reliability of downstream analysis. Okay, so mainly this tool can handle different types of sequencing data, including Illumina. Okay. Illumina and pad and single and Solaxer data. It also offers several trimming and filtering options like trimming by quality, removing adapters and contaminants, and removing reads that are too short or too long. So here we can search Trimomatic here again. Trimomatic. Okay, so click here. Okay, so here you can see page like this. So here our data is paired and right. So here if your data is a single end, you can choose single end, and our because right now we are using paired and data, so we had to choose paired end here. Okay, then you can add the result. I mean here, then you can choose here like fast QC. Okay, you can select here control first and this first. This is R1. Okay, so select 1 and 1. Okay, then again select R2 second pair. Click here. Let's see this first. Okay, then okay. Then again you can here you can add same default parameters. Okay, then run tool. It's running now. Yeah, so here also it is started the the automatic tool successfully added the sequence, and here it's running now. So it's also in queue. It also will take time. Okay, so here I'll show my automatic result. So it will be like this. The result will be like this. Okay. Okay, I'll show the result here. So it will be like FASTQ format. Okay. So trimming is done actually. So we have removed the low quality reads. Then next we are going for aligning or genome indexing. So for that we are using HiSat tool, HiSat 2. HISAT. So you also do with me. Otherwise, no, you'll get confusion. Confusion. So both the tool you already done, right? And Trimomatic. Trimomatic also you do. You did, right?
Yes, ma'am.
Okay, so then you just search for high sat tool. So here for aligning also we will be getting so many tool. So usually for RNA-seq analysis we are using star and high sat. Okay, so star means so many tools are maybe you may heard like Bouty one, Star, HiSat, and so many tools. Maybe you heard about that. So mostly Bouty and Bouty one, it will be using for that variant analysis, ChIP-seq analysis, that kind of things. And for RNA-seq analysis, mostly we are using for aligning purposes. Mostly we are using star and HiSat. Okay, so our star is very good tool, and even HiSat also. But HiSat star will be taking much memory than HiSat, and it will be taking more time. So it will take 1 day or 2 day to complete, and it's depend upon that reference genome size. So HiSat means it will take compared to star, it will be very fast. So mostly for me it took I think 20-30 minutes. That's it. So here we are doing HiSat now. Again use just HiSat. This is the HiSat tool. Okay, click here. So here you can see this page, HiSat tool page. So HiSat means it's a it's using for hierarchical indexing for spliced alignment or transcript. Okay, it's very useful for quick and accurate alignment sequencing, mainly RNA sequencing data. It is an extension of HiSat and uses a hierarchical indexing strategy based on the Gauss-Wheeler transform and Faragina-Mancini index to efficiently align. These are the algorithm they are using and to efficiently align RNA secretes to reference genome or transcriptome. And mainly it is very capable of identifying known and novel splice sites and can align reads across exon-exon junctions, and this is important for accurate gene expression quantification and transcript assembly. So here we are. I think alignment means you all get it right. Alignments where we are aligning our reads to a reference genome. Okay. So here already built-in genomes are available in Galaxy. So we don't want to download a separate genome. So here we can select use a built-in genome. Then here, since it is a human sequence, so here we are going for a human reference genome. Okay, select human, this one. Okay, this is the human reference genome we are using here, SG38.
Ma'am,
Okay.
ma'am, actually here we selected humans, so human reference is there. If in this platform plants also it will be there, ma'am.
Yeah, plants also it will be there. If it is not available, you can go for genome assembly. Okay. Otherwise, some other technique also available now. I mean, simulation techniques also available. So that bit difficult to understand. I think you can read some papers like regarding simulation. So it will build automatically like genome, like artificial genome. Then it will compare. It will align normally. So that type of paper you can read like simulation technique. Otherwise, you can go for genome assembly.
Okay.
Assembly. Okay. So here, okay, we selected use a built-in genome. I
Okay,
mean,
ma'am.
already selected a genome for comparison. Then we can select here single and or parent. So our is like parent. Yeah, parent data we selected. Then again here trigonometric finished, right? So here you can select pad sequences. Okay, so this is like okay, R1 pad. Okay, here I am selecting R1 pad. Then it's taking time for me. Yeah, yeah, here R1 pad. Yeah. Oh, it's here I'm selecting. I think we got. I'm deleting is this okay to Matic? I will be confused. First is also I'll delete. Mm. Oh, physical also deleted. Oh, okay. Then you can select here multiple. Then you can select a pad sequences, R1 pad. First, R1 pad sequences. R1 pad. Oh, twice. Okay, R1 pad. Okay, here, R1 pad. Two sequences it will be there. Then again here, second option. Okay, second thing. So second, here you can select second pair like R2 pair. Okay, R2 pair, and here also you can R2 pair. Okay. Then you can go this one, you can select as forward read. Then here you can select parent option. Parent option specify parent parameter. Here you can select this option here, and then other thing it's like make it as default. Okay. Then run the
ടി വി
tool.
ഒന്നും ഇടല്ലേ, ടി വി ഇടല്ലേ. എൻ്റെ നെറ്റ് ഉപയോഗിക്കുന്നത് നോക്കിക്കോണേ. എൻ്റെ നെറ്റിൽ മൊബൈലിലാണ് ഇപ്പോൾ ഓടുന്നത്, നെറ്റ് കിട്ടുന്നില്ല.
So here again you will get a green color here. So it's added here. So high set. Here you will get like this high set and
paired and option is use default values.
pad and no pad and I selected right now. That also you can default, but mostly it won't get
അതൊന്നും കുഴപ്പമില്ല.
affected. But you can
എൻ്റെ
select
അങ്ങോട്ട്
that.
ഓൺലൈൻ
Whatever I selected, you can select that also.
Which one? There are two options: use default values or specify paired and parameters.
Pad and parameters I selected right.
Oh.
This one parent, where it is like high set. No high set. You selected right here. Pattern. Then you can select here your sequence like first pair you can select here and second pair you can select here. Then yeah, then you can select a forward route since it is yeah. Then you can specify pattern parameters. So you can select this
Forward.
option. Yeah, forward.
Okay.
Then you can go other thing you can set as a default parameters. Then you can run the tool. So it will be like it. You will get a page. I mean, you will get a queue like this, like two things. So it will show in history lane. Okay, so it will be like you'll get a result like this. 1 minute, where I got the result. Yeah, it will be like this. Yeah, you will get a result like this. Okay, so it's a BAM file actually. Okay, then aligning is done. So after aligning, we had to quantify the reads. Okay, so for that we are using feature counts tool. So feature counts. Okay, here you can select feature counts. Counts. Here, feature counts measure gene expression in RNA-seq experiments from SAM or BAM file. Since it is BAM file, so we are using feature counts here. So it is mainly okay. You can click here. Okay, so it is a very highly efficient general purpose and read summarization program that counts mapped reads for
Hello. Hello.
I think ma'am having a network issue. There is no voice, I think so.
Yes.
I'm not hearing her voice.
Yes, yes. I thought I have a issue. That's why not responded. So Dr. Vijay has some problem. I think she will join. Please be there. So
Yes, sir, ma'am. I think ma'am left. I
yes,
think
yes,
so.
yes, yes. Hope it is going okay for everyone. The workshop is going. If you have any doubt,
Yes.
if you have any doubt,
Yes,
you
sir.
can
It's going fine, sir.
switch on your mic and ask. I think it will go one more day as per Dr. Vijay's plan. So everybody is okay with the Saturday or Sunday. I think some people have problem with Saturday. Can we shift to Sunday? I think Dr. Vijayana will
Yes,
tell
sir.
you. Sunday the
Yeah, yes, sir.
same time.
Yeah.
I think Dr. Vijayana will discuss with you the time everything. But I think we can do one more session on Sunday. But she has planned to complete some more tool, so I think she will come back. So
Yes, sir.
so hope you are following. That's what
Yes,
I
sir.
meant.
Sir, I am having one small doubt, sir.
Yes.
In future, if you can arrange Dino assembly also, it will be useful for us also, sir.
Yes, yes, yes.
Because,
We will.
because ma'am told some reference genomes will not be there in that software, so
Okay.
you have to go for Dino Wonder. If we
Yes,
are
yes.
exposure to that also, it will be useful for us for our research, sir.
Yes, yes, yes. We will conduct. We will conduct.
Okay, sir.
We need to find out some person who are well versed to that, and then we will conduct. I think if Dr.
Okay.
Vijay is okay with that, we will conduct with her. Otherwise, we will find out some person and conduct. Okay.
Yes, sir. Yeah, sure. Sir.